So in this chapter, we are on chapter 4. We're looking at carbon and the molecular diversity of life. So carbon is essential to life on this planet. All life forms are made up mostly of carbon. So organic chemistry is the branch of chemistry that specializes in studying carbon compounds. Sometimes you will hear it referred to as the chemistry of carbon or hydrocarbons. So previously, they had more of a vitalistic belief that there was a life force outside the jurisdiction of the physical and chemical laws. Basically, that anything that was alive was governed by different rules than the rules that applied to anything that was not alive on this planet. This was a foundation for the new discipline of organic chemistry. This foundation was chipped away as we learned to make organic compounds in the lab. So the first organic compound they made in the lab was urea, and they made it without a kidney. At that point, they started to look at things and question it and say, okay, maybe these physical and chemical laws are going to govern all natural phenomena, including the life processes. And this is more of a mechanistic view that we have today. So carbon forms several diverse molecules by bonding to 4 other atoms. So we know that carbon structure has 6 electrons. 2 of them are in the first shell, 4 are in the second shell. So it has 4 valence electrons and can form 4 covalent bonds so that it has 8 electrons present in that shell. It will form a tetrahedron shape with angles of 109.5 degrees when it forms 4 single covalent bonds. So naming of the carbon compounds is really pretty straightforward. A lot of times the names can be intimidating to people when they are first learning them, but with being aware of just a couple things in here, it can make it a little bit simpler and not quite as intimidating to look at. So first thing we look at is the number of carbons in the chain. So if there's 1 carbon, it's going to use the prefix meth. If there's 2, you use the prefix F. 3, you use the prefix prop. 4, you use the prefix but. Then we look at the number of bonds. If there's single bonds, you're going to use an ane ending. So for example, 1 carbon, all single bonds, we call it methane. If there's double bonds, we use the ene ending. If there's triple bonds, we use the yne ending. So for example here, ethane is going to have single bonds, so we use the ane ending, and it's got 2 carbons, so we use the F prefix, ethane. If you have a double bond in there, now it becomes ethene, or sometimes also called ethylene. So when we look at the diversity of carbon, there is quite a wide range of molecules that can be formed. The carbon chain is going to form the skeleton of most organic compounds, and it can vary in length. It may be straight or branched, or in rings. It can vary in the number of bonds, and then additional elements can bond to the skeleton. So if we look here, it just changes in the length. 2 carbons versus 3 carbons, we have ethane and propane. We can look at where the double bond position is. So here, the double bond is at carbon 1, so we call it 1-butene. Here it's at carbon 2, so we call it 2-butene. So the number 2 is just specifying where the double bonds are. When you see the numbers in the names, they give you a lot of hints, and do not be intimidated by them. When something is branching, so here we have butane that has 4 carbons. If it's branched, we can use the name isobutane is one option, where it's an alternate form, or we can use the name 2-methylpropane. So propane is the main skeleton. At carbon number 2, we have a methyl group. If there's rings, you can have cyclohexane, for example, 6 carbons to give you the hex, all single bonds, the ane ending, and cyclo because it's round. Benzene is another example. Here, every other bond is a double bond, and you've got 6 carbons. So hydrocarbons consist only of carbon and hydrogen. They're a major component of petroleum because partially decomposed organisms are going to contain a lot of hydrocarbons. They're not prevalent in living organisms, but many of the molecules have regions that are hydrophobic or undergo reactions that release relatively large amounts of energy. So one of the reasons that we burn petroleum products is there is relatively large amounts of energy held in those hydrocarbon bonds. So we get a lot of heat for the amount of substance that we're burning. So your isomers are the variations in the architecture of the organic molecules. We can have different types of isomers here. So the first is the structural isomer. These are going to differ in their covalent arrangements of the atoms. They have different neighbors. So if we look here, all 5 of these carbons are a ring in this format. Here they have different neighbors because one of them is off to the side. So here nobody has more than 2 carbon neighbors. This one actually has 3 carbon neighbors. So they're different in their covalent arrangements. Geometric isomers, they're going to have the same covalent partners, but they will differ in special arrangements. The example would be our cis and trans isomers. So here when you have a cis isomer, you have 2 of element X on the same side. When it's in the trans formation, the 2 element X's are on opposite sides of the molecule. So you may have seen this terminology used if you look at food lately. One of the things that people have become aware of is the health concerns associated with trans fats. So when they're talking about trans fats, they're talking about things being on opposite sides of the molecule. And it will have different chemical properties when it's on opposite sides than if it's on the same side, and it may need different enzymes to interact. So your enantiomers, these are isomers that are mirror images of each other. So your middle carbon is going to be asymmetrical because it's going to be attached to 4 different groups. You can almost think of these as being left and right-handed versions. With these, usually one molecule is going to be biologically active and the other is inactive. We found this out when we looked at some of the drugs in the past. So the enantiomer of a drug may not be equally effective or even safe. So our classic example of learning about this in the drug industry was the drug thalidomide. So the good enantiomer had the desired effect. The other enantiomer had a harmful effect. So this drug was used for morning sickness and given to pregnant women. The good enantiomer helped with the morning sickness. The bad enantiomer was converted in the patient's body, and it was actually a very strong teratogen. Teratogens cause birth defects. So we were giving these to pregnant women that would, knowing they were pregnant, and it would cause them to have birth defects. So the drug was pulled off the market some time ago, and then it was actually released back on the market because we found out that it actually can be helpful for some other patients out there. So it can be used for cancer patients. If a patient is on thalidomide now, birth control is mandatory. So they want to absolutely ensure that nobody taking this drug will be pregnant. So this picture here shows your L and D isomers, which are like your left and right-handed isomers. So if you look, it's actually how it spins light is how these are looked at. So then we have these chemical groups that are going to be helpful in understanding the function of biological molecules. We call them the functional groups. They're a chemical group that will replace one or more of the hydrogens bonded to the carbon skeleton. The functional groups can participate in the chemical reactions in a characteristic way. These are 7 functional groups we use in biology. There are certainly more than this in chemistry, but what we're concerned about are these ones that are seen in biology today. So the hydrophilic groups, these can be used to increase water solubility. These include the hydroxyl, carbonyl, aminos, and sulfhydryl and phosphate groups. Methyl groups, these aren't very reactive, but they do put a recognizable tag on biological molecules. So we can use these when we're trying to control transcription or translation, turning on and off a gene. So this is a simple table here that is typed. This is also going to have some examples that show the chemical structure down here. So you can refer to whichever one you would like. The hydroxyl group is going to have this oxygen and hydrogen that is going to be attached. These are the alcohols. An example would be ethanol. These are going to have polar bonds. So the hydrogen bonds with this can help dissolve organic compounds. This is something that the liver would use in trying to help to break things down and flush out toxic things in the body. It's a way to help make them more water soluble so that it can be flushed out through the kidneys. The carbonyl, here you're going to have a carbon with a double bond on an oxygen to one side, and then the carbon attaches to something else on the other side. It's called a ketone if it's within the skeleton, and it's an aldehyde if it's on the end of the skeleton. So here down below you can see one that is on the end of the skeleton and one that's in the middle of the skeleton. So these will make your aldose and ketose sugars. The carboxyl group, this is going to have a carbon double bonded to an oxygen on one side and then a single bond to an oxygen and a hydrogen on the other side. These are going to be used to make your carboxylic acids or organic acids. An example would be acetic acid, which is vinegar. This can give acidic properties and be found in cells. Your amino group makes the amines. With this one, you've got a nitrogen bonded to two hydrogens. An example would be glycine. These can act as a base. They're ionized with a +1 charge under cellular conditions. Your sulfhydryl group is going to have a sulfur and a hydrogen. These are the thiols. An example would be the amino acid cysteine that would contain some of this. The cross-linking between the sulfhydryl groups can provide really strong bonds. You can find this in some of your harder tissues, and it can determine the curliness or straightness of hair. So if you were around in the 80s and you got perms in your hair, you remember that they have a lovely smell to them. Or if you're around some of the elderly ladies, they still seem to like the perms quite a bit. You can get a chance to get an idea of what the thiols will smell like. So your phosphate groups, these are going to make up your organic phosphates. Example, glycerol phosphate. This is going to contribute to the negative charge of the molecules. These phosphate bonds also have the potential to react with water, and they can hold a lot of energy and release a lot of energy in the bonds. So down here you can see an example of a phosphate group. And then last we have the methyl group. It's a carbon with 3 hydrogens attached. This makes your methylated compounds. An example would be 5-methylcytidine. These can affect gene expression and shape, the function of the sex organs. When you're affecting gene expression, you have a huge role in the body. So that is chapter 4.